This is question 3, and we've got a section through a human ovary. And name the structures. And so A is pointing to these cells around the Graafian follicle. And so you can say sort of any of the cells that you've heard of, thecal cells. Even if you call them follicle cells, you can get away with that, or granulosa cells. So these are theca cells, or you could say follicle cells, or you could even say granulosa. And then B is pointing to, it's got, the label has got kind of a large bar on the end, so it's saying this whole thing. And so that is the smaller one, should give it away, is the primary follicle containing the primary oocyte. So primary follicle. And you could possibly get a mark for primary oocyte, but it's not really pointing to that, it's pointing to the whole thing. And then we've got 3 marks worth of maths, saying the volume of our primary follicle, B, is a very small number in millimetres, and the volume of a mature Graafian follicle, this big thing here, is this bigger number. By how many times bigger is it? So we've got a calculation, and then we've got to convert it into standard form. So again, if we're doing any sort of magnification calculations, it's always the same. So take the big thing, and our big number is 4.19 times by 10 to the 3. We don't need to do any conversions in millimetres, they're both in millimetres. And so how many times does this other number, 0.4 zeros, 1, 2, 3, 4, and then 5. So how many times does our primary follicle go into our Graafian follicle? And then we're going to calculate that and then just convert that into standard form. So with your calculator, you just need to go 4.19, and however you do exponentials on your calculator, 4.19 times 10 to the 3 divided by this 0.00005 equals 838, and then you got 1, 2, 3, 4, 5 zeros on it. 1, 2, 3, 4, 5. So that's 83 million times bigger. But it wants it in standard form. So standard form is a number between 1 and 10. So in this it's going to be 8.38. And then how many times you move the decimal place. So 1, 2, 3, 4, 5, 6, 7. It's going to be times by 10 to the 7. It doesn't have any units because it's just how many times bigger. Some of you might have calculators that do that automatically. So there's your 3 marks. Then on the right we've got first meiotic division happening just before ovulation. Rather gruesome. It's even worse in colour, this picture. And there's your Graafian follicle. So you can actually see how it's quite big. Structure X contains the secondary oocyte. There it is on the left. Name the 2 outer layers. So this is just remembering the names of them. So strange names: Zona pellucida, and the other one Corona radiata. So just AO1 stuff there. And likewise AO1 for describing how the sperm penetrates the outer layers of this structure. So it's this acrosome reaction, so just mention acrosome and then just giving us a bit more detail about that. Releases enzymes which hydrolyze these layers or digest the layers. So releases enzymes that hydrolyze anything that's going through layers, or you could say digest layers. Then we've got part D about IVF. It tells you a bit about IVF because we don't really do that much stuff on IVF in our syllabus. And secondary oocytes are removed and are fertilised with sperm in a lab. That's what test tube babies. And there's a risk that more than one sperm enters a secondary oocyte. So this would occur possibly naturally, but there's an increased risk if it's not fully mature. Okay, and the 6% increase in risk. So with IVF, you tend to get more twins. State how the secondary oocyte usually prevents the entry of more than one sperm, and suggest a reason why this is increased with IVF. So there's two things you need to do clearly in the question, and there's 2 marks available. So we know how to structure this now. So the thing that normally happens is this layer around the oocyte, once it's fertilized, stops any further sperm from going through, and we just need to remember what exactly happens to what named this sort of cortical reaction. So let's say cortical reaction. So cortical reaction, and the zona pellucida thickens, or sometimes called a fertilization membrane. So zona pellucida thickens, or words to that effect, or a fertilization membrane forms, or fertilization membrane. So that's what normally happens. And with IVF, it does say that a secondary oocyte is not fully mature. So that could be a problem with this reaction. So maybe cannot form this membrane, or it cannot thicken. So with IVF, possibly zona pellucida cannot thicken. This fertilization membrane might not form because of less of those cortical granules, so less cortical granules. So what normally happens, and then what we're at risk of not happening. In part 2, the developing embryo is not transferred into the uterus until 3 days after the fertilization in the lab. Using your knowledge of fertilization, there's a key clue what you need to do, and implantation, explain why this delay is needed. So let's trawl our knowledge of those and see if we can get 3 marks of it. So our knowledge of fertilization. Where does fertilization normally happen? It happens in the fallopian tubes. So fertilization normally happens in the fallopian tubes or oviducts. So that's our knowledge of fertilization. And then, if you remember your geography of the female, then that's a little distance away from where implantation is going to take place in the uterus. So you've got a kind of uterus, and then you've got fallopian tubes, and so we're excellent diagram I know. But if fertilization takes place there, then there's a little bit of time before implantation in the endometrium there. And in that time, the zygote, so the fertilized oocyte egg, has a chance to turn into a blastocyst of about 100 cells, and that enables it to do all sorts of stuff to make sure that it can embed in this wall. So that's what you need to sort of put together. So the embryo would normally need to reach the uterus and form a blastocyst. So embryo needs to form the blastocyst, or that kind of cell cleavage take place. So that's what normally happens. And in that sort of normal time, in the cycle, then this lining would be getting thicker. I'm not convinced that in the IVF the timing is always, they take the egg, fertilize it, and actually in the same cycle. So if you see the mark scheme to this, I'm not convinced completely it's exactly what they do. But we need another mark to sort of go, well, what would happen to this blastocyst? So the endometrium needs to be kind of fully developed. So we can certainly put that, because we're talking about implantation. So endometrium, or the uterus lining, needs to be fully developed. And so it wouldn't be if you put it in too early. And other things we could say, needs to form these structures, the sort of connections between the uterus and the blastocyst, the chorionic villi. Chorionic, so this is all from your notes. Villi. And really you could ignore that and leap straight to the end. If this blastocyst is put in too early, then, or if it's put in before it's a blastocyst, then too early in the stage of development, it won't implant and won't survive. So embryo would not implant, and so it wouldn't survive. It would just go out, through the vagina. So implant or survive. So the key points there are: tell us what normally happens in the fertilization. So normal fertilization, turning into a blastocyst. Maybe mention this implantation. So we need a thickened endometrium, and the risk with IVF of losing the embryo.